So when we talk about looking at molecular signature selection, we basically then have to go down and talk about some basic features of population genetics. What I want to basically do is review some of the structure things you expect to see with neutral variation. Neutral variation means that I've got drift causing allele frequencies to randomly change. And remember, what drift does is it eventually causes all alleles to be lost. Then I've got mutation introducing new variation. The difference we talked about before is I'm not worrying about the effect of a new mutation. I'm simply looking at sequences and asking, how many alleles do I have? What's their frequencies? In quantitative genetics, we also have to worry about what's the effect of an allele. Here, we're not worrying about that. So as we move from that's great wording, isn't it? As we move to tests based on molecular markers, man, don't drink when you write lecture notes. I don't drink. Yeah, it still doesn't matter. I'm dyslexic, so I make all sorts of really interesting errors that, you know, they're really fun to catch. I'm sure some really great errors, you know, stupid typos like that embedded in the book despite our best efforts. So as we move to tests based upon molecular markers, either in a candidate region, so I specify a gene, for example, I think hemoglobin beta is under selection, or a genomic scan. I have no idea what's going on, let's scan the entire genome. And historically, the tests have kind of moved from locus specific, because they were hard to get information on a lot of stuff, to now genome-wide scans. These are trait independent. I'm not using any information about the trait. That's a strength and a weakness. If I see a whopping big selection signal on a chromosome, it's often very difficult to turn that into what trait was under selection. So these are complementary approaches, trait-based approaches and marker-based approaches. So what we use here is allele or gamete frequencies. We don't use additive variation. So we simply, if I have a DNA sequence, I have no information about what the phenotypic effect of that DNA sequence is. Because I know nothing about its effect size. So to do these tests, we need an understanding of the pattern expected for strictly neutral alleles. So the neutral equilibrium model, also called the standard equilibrium model, is, think about a swimming pool. You've got a drain in a swimming pool, and you've got water coming in. That drain is drift. If that drain's open really big, the population size is small, drift is very good at removing variation. Water coming in is new mutation. Eventually you'll get to some equilibrium level where the water coming in, the water going out balance, and the pool's at a certain height. That's your level of variation. However, the water coming in is new mutations, so it's changing color all the time. Why? Because you have new mutations being introduced. So over time, even though the level of water in that pool is roughly constant, the amount of variation is roughly constant, the composition, the color of that's going to change. So under drift and mutation, you get some equilibrium level of variation plus noise about it, but over time, populations diverge. Why? Because new alleles arise by mutation and old alleles are lost by drift. So two identical populations will diverge over time. So under the standard the neutral equilibrium model, we assume strictly neutral alleles, and we assume the population is in mutation drift equilibrium. Think about a pool, you have an adjustable drain. If right now I take that pool and adjust the drain, it's not in equilibrium. It hasn't run long enough. If a population goes through a bottleneck, if you don't wait for a sufficient amount of time, it's not in equilibrium. And this is a critical feature because almost all tests of selection based upon markers, when they fail, that is, you reject the neutrality test, you could reject it because you don't have neutral alleles, or you could reject it because you're not in mutation drift equilibrium. And this is the key issue, because most populations are not in mutation drift equilibrium. They've gone through bottlenecks recently. They've gone through expansions recently. They have population structure. Most populations have changed. The populations people typically look at, you know, until quite recently, the model systems people follow are humans, Drosophila, Arabidopsis. We can apply all this machinery to non-model systems without any changes, but those are the big three people following. And all three of those populations have undergone massive expansions. Humans and their commensal Drosophila expanded out of Africa in a series of migrations through Europe, North Asia, South Asia. Arabidopsis basically experienced population expansion following the retreat of the glaciers. So the big the big organisms people have done multiple tests for have situations where you expect them to be out of mutation drift equilibrium. So all the tests people use, almost every single one of them, you then have to somehow adjust for, what if I'm not at equilibrium? So the key part to take in mind is that the population has to be constant for a sufficiently long amount of time to reach mutation drift equilibrium, and that's a tough assumption to make. Also, no population structure. So what's the behavior of I introduce a new allele, what's the behavior under drift alone, simply drift? Well, it results in one lineage ultimately being fixed. This is coalescent theory. Let's frame it this way. There's 50 people in the room here. You have two alleles. Let's number every allele. I don't care if they're the same or not. So I've got numbers 1 through 100. We can think of those as a lineage. What are the descendants of allele numbered 1? What are the descendants of allele number 27? If I run drift long enough, eventually everyone in the room here will have an allele that descends from a single one of the copies that's here. If I go to a different gene, I see the same thing, but I have a different, traces back different copies. So for example, all the alleles in 400 generations may trace back to my first copy of allele 1 for locus 1. They may trace back to your first copy for locus 7 and your first copy for locus 10. So we're a chimera of different ancestors and lineages. So one way to think about drift is think about lineages and loss of lineages. So if I have no new variation being pumped in, if all alleles eventually trace back to a common ancestor and there's no mutation, I lose variation. Also, an important part we'll talk about is the neutral allele frequency as a function of age. A common allele under neutrality is an old allele. And what do you expect for an old allele? Well, if I have an allele that has a large block of linkage to disequilibrium about it initially, as it gets older, what's going to happen? That linkage to disequilibrium block is going to decay by recombination. So an old allele should have a small amount of LD around it. Therefore, common alleles should have small amounts of LD around it. There's a large class of tests we'll talk about probably on Wednesday called long haplotypes. And that is alleles which are common, which have long blocks of LD. That's the signal you usually don't expect under population structure or departures from equilibrium. And the only way you can get a common allele that seems to be young, the large block of LD is young, is usually by selection. There's one other feature called allelic surfing we'll talk about that can do that as well. So the point is, there are a bunch of features we're going to exploit here. And again, what I want to stress is this lecture and the next lecture are kind of to set the groundwork. Then we're going to go back and drill down and examine all these points in detail. Okay? We've seen these before about the loss of variation. The loss of variation, there's the heterozygosity at time t, heterozygosity at time 0. Loss variation depends upon the effective population size. I just put this up there for fun. You can actually solve what the expected allele frequency is under drift. So if you start out at frequency p0, what's the expected frequency at time t? You can actually solve for it using something called diffusion theory. It's a set of approximations. And you can actually write down an expression for it. And for example, here's what happens. If you start a population initially at frequency 1/2, you can then look at this two different ways. Number one is imagine 1,000 replicate populations. This would be the histogram over time. Number two, if I ran that same population over and over again independently, this would be the histogram. So initially, everything is at 1/2. At time t, so 1/10 of n, I get kind of a bell-shaped spread about that. And what you basically see is as time gets going down here, time is getting larger and larger and larger. It gets flatter and flatter and flatter. Eventually you get this interesting expression where the allele frequency for those which are still segregating is independent of its starting condition. If I start at a more extreme value, here, I think it's at 1%, what you basically see is that among those alleles that are segregating, if you wait long enough, again, you get this flat distribution. So we have a very good idea about how neutral alleles behave in a large, constant size, finite population. We can write expressions down for mean time to loss, mean time to fixation, and basically time the allele hangs around in the population. We'll occasionally use these a little bit in tests for selection. And basically the idea here is here's the time to loss, here's the time to fixation. And the key part gets to this notion I mentioned that a common allele is an old allele. So you can basically ask, if I introduce an allele as a single copy, under drift alone, if I tell you its frequency now, how old is it? There's actually an expression for it. If this is its current frequency, this is the expected age of that allele. So it depends upon the effective population size and its frequency. This, by the way, is an expected value. That's an average value. It's got a huge variance, an enormously large variance. How can we apply that? Well, here's one of the early applications. So a deletion, del32, in the CCR5 receptor, that's the receptor that HIV uses to get into the cell. If you have this mutation, you're largely resistant to HIV infections. This mutation, this deletion, occurs at about a 15% frequency in Europeans and Asians, but is absent in Africans, Native Americans, and East Asians. So if you assume a frequency of about 10% and an effective population size of about 5,000, then when you plug into this expression here, it says the age of that allele should be about 5,000 generations with some pretty big standard error on that. So an obvious question to ask is this allele too frequent relative to chance? That is its frequency too high given its expected age? To do that, you need some other approximation of its age. And we'll come back to how you can get that. But one way to get an approximation of an age is suppose here's the allele, and that allele arose on a single chromosome. Let's imagine it arose with this other nearby linked allele. So when this allele originally arose, it was entirely on this chromosome. We can then age that allele by asking what fraction of chromosomes now contain this, because over time they're going to diverge by recombination. How do we know it was on this background? Because it's the highest frequency one. So an independent estimate of age is what fraction of the original combination is maintained. If I know the recombination frequency, I can compute that. When you do that for this example here, as we'll talk about in a second, your estimate is on the order of about 20 generations. So this allele is too frequent relative to what it should be given its age. That is, the allele is an independent estimate of its age shows that its age is much younger than its frequency should be. It's at too high a frequency for its age. It's probably been driven up there by selection. So again, this is a little bit choppy because I'm trying to set some basic general ideas. We'll come back and talk about these in detail. What I want to talk about in a fair amount of detail is the coalescence structure. And the coalescence structure has to do with, as we go either back in time or forward in time, what happens to genealogies. So I look at everyone in the room here. I've got 100 different alleles. If I go back in time, it may turn out that 5 generations in the past, a copy of one of my allele and a copy of one of your alleles came from a common ancestor. So I move from 100 different lineages. A couple of generations back, I now have 99 different lineages. Why? Because my allele, one of my alleles, and one of your alleles had a common ancestor. So I go back 4 generations. Those two have coalesced, and I now have 4 generations in the past, 99 different lineages. I can go back 98, 97. Eventually, they'll all coalesce to a single point, and that's about 4n generations ago. And that branching pattern of ancestry is called the coalescence structure, and that's a critical part to basically ask how selection impacts things. So let's look at that in a little bit more detail here. Here's a coalescence structure. So basically, here are 5 alleles I've sequenced right now. If I go far enough back in time, they all come from a common ancestor, but there's a branching pattern. So for example, how long do I have to go back to go from 5 independent lineages to have 4 independent lineages? Well, it's actually pretty straightforward. The chance that any 2 alleles have a common ancestor is 1 over 2n any given generation. And I basically have 5 choose 2 different ways to get that. So what I can basically show is that expected time is given by this here. So if I have n to go to n minus 1, it's 4n, and this is the number of different combinations I have. So for example, for 5 alleles, on average, it takes about 0.2n for you to have the first coalescent event, about 0.33n for the second, about 0.67n for the third, and you'll notice here that most of the time is for these last 2 to coalesce. So the entire population has a coalescence that's on the order of about 4n, but most of that time is with the last 2 things coalescing. This is the structure you expect under neutrality. When you have selection, number 1, this thing gets squashed down. Number 2, the distance between these nodes gets compressed. And as we'll see, that actually impacts the molecular signature. So one way to think about the importance of a coalescence is, this is the genealogical pattern I expect. I now throw down random mutations. If I throw a random mutation and it lands here, it means these 2 individuals have it. If I throw a random mutation and it lands there, it means only that individual has it. If I throw a random mutation and it ends there, it means 3 individuals have it. So once you have the coalescence structure, it basically tells you about how many of the individuals are going to share it. So when you change that coalescence structure, you change the distribution of allele frequencies. And there's a large number of tests that basically ask, are the distribution of allele frequencies down here consistent with this kind of coalescence structure under drift? Because that coalescence structure gets significantly modified when you have selection. We'll talk about that in a lot of detail. That's why a lot of tests for selection are based upon this idea about coalescent theory. So again, this is just kind of the branching, the genealogy. I have 5 alleles down here. How far do I have to go back until I only have 4 lineages, 3 lineages, 2 lineages, 1 lineage? And the key part is under drift. That's entirely determined by the effective population size and the number of lineages. There's a very nice clean pattern for that. That gives us an expectation. We can then contrast that expectation with what we actually see. So I'm trying to walk a fine line here between kind of trying to show you where some of this stuff comes from at kind of a heuristic level without actually drilling down and going into the hardcore mathematics of it. And I worry by doing that, I kind of disenfranchise both groups, right? The thing about the coalescence structure basically is that geometry, when you throw a random mutation down, gives a specific pattern. If you perturb that geometry and then look at the mutations down here, you'll get a different pattern. And a lot of tests basically have looked and asked, is the pattern I see down here of allele frequencies, is that perturbed relative to what I expect under drift? And so things like Tajima's D and other tests you may have heard about, the singleton density score, are all based upon this general idea. So don't worry about the details, the numbers, just think about that basic idea. If I've got a certain geometry and I throw 5 mutations down at random and do it lots and lots of times, I expect a certain pattern. If I change that geometry and again throw 5 mutations down, I'm going to get a different pattern of allele frequencies down here. And tests of selection basically look at that different pattern of allele frequencies and ask, is it sufficiently different from what you expect under drift? Okay. And we'll talk about that a lot, but that's where that comes from. Okay. So if you get the rough idea, that's the important thing. Or you can spend a lot of time and dig through the coalescent theory and get the exact idea. So I think the rough idea is pretty good. So a key here is a magical parameter called theta. If I'm at mutation drift equilibrium, a huge amount of detail about my problem, my population, in fact, essentially everything, is given by two quantities. Number one, 4 times the effective population size times the mutation rate. Number two, my sample size. If I give you those two, I can determine a huge number of different things under drift. So for example, if I have exactly k alleles that can arise, and a special case of k is a SNP, where I've got 4 potential alleles, the equilibrium heterozygosity, that is, with the chance I draw a random individual out, it's a heterozygote, is given by theta over this thing. Theta is usually pretty small, so to first approximation, all these things are basically theta. The infinite alleles model is a classic model that says each new mutation is unique. Think about a DNA sequence. And again, the key part is in basically all these models, theta is usually small, usually much less than 1. So these things at the bottom don't matter much. And no matter what your mutational model is, at equilibrium, your heritability, your heterozygosity is roughly given by this thing called theta. So if I take a sample, so each of you is an individual. I take a DNA sample from you, and I want to ask at a given locus, is there something about that DNA sample from you that suggests departures from neutrality? Well, there's lots of things I can measure in that sample. I can measure the heterozygosity. What fraction of your loci are heterozygote? I can measure a very close related thing called the nucleotide diversity, right? If I look at the entire population, what fraction of sites show variation? I could measure the number of singletons. How many sites I look at, I've got an allele is found in only one individual, a singleton. So think about a SNP. Everyone here is a G, but one individual is an AG heterozygote. That's a singleton allele. I then look at allele frequencies. What's the frequency, and the way to look at allele frequency is something called the frequency spectrum. So we'll talk about site frequency spectrum and allele frequency spectrums. I could ask about the allele frequency at a given locus. A frequency spectrum says, look over all loci and what's the distribution of allele frequencies. So suppose I've got M sequences, and in those M sequences, I look at a number of different loci. Then for that set, I compute this statistic here. N1, N2, up to Mn, Mnm, where Nk is the number of alleles present as exactly k copies. So for example, if my allele is monomorphic, I look at a certain SNP, everybody's a homozygote for that SNP, then the number of, there's one time that's present where I have exactly M copies. If everyone is a singleton, that is, I look at that locus and basically everybody's got a different copy, then I've got M times it appears as only one allele. Suppose I have 10 sequences, I've got 4 singletons, one present is 2 copies, one present is 4 copies, then the site frequency spectrum would say 4 times it appears as once, 1 time it appears as 2, 4 times it appears as 1, all other combination appears as 0. And if you think about it, you do k times n plus k, that gives you back n. So it's kind of a weird notation, but it's a compact way of representing how often different sequences are. You have nothing but singletons, you've got 1 copy, 2 copy. Basically, it's a complete way of looking at the allele frequency over a large number of sites. And it's either called the site frequency spectrum or an allele frequency spectrum, depending upon if you think of these as sites or alleles. We'll talk about that distinction in a second. So when you analyze DNA sequence data, you basically have 2 general frameworks. You can have a so-called infinite sites. I think nowadays it's called infinitely many sites. Or you could have an infinite alleles framework. And the distinction is under the infinite alleles framework, you take a block of DNA that you assume has no recombinations in your sample. So take it small enough, it'll have that. Each different DNA sequence is a different allele. This requires phase data. And so the allele frequency spectrum will be how many different haplotypes do you have? How many haplotypes are present? There's 1 copy, 2 copies, 6 copies, etc. That's infinite alleles. Infinite sites, infinitely many sites. I'll show a picture of this in a second. Each nucleotide is considered as a different site. Again, I'm assuming no recombination. This doesn't require phase data, but it may require, depending upon your model, polarized data. So by that, here's an example. So here are 1, 2, 3, 4, 5 sequences. To make it easy to visualize, I've underlined the variation here. So how many alleles do I have? That's 1 allele, that's a 2nd allele, that's a 3rd allele, that's a 4th allele. No, so I have 1 allele, so I've got 1 allele, 2 alleles, so I've got 3 alleles in here, right? So if I take this DNA data and treat it as haplotypes, I've got 3 haplotypes. That's analyzing it in an infinite alleles framework. It requires you to know the haplotypes, to phase the data. Infinite sites model, look down there, that's 1 site, it's monomorphic. That's a 2nd site, it's monomorphic. That's a 3rd site, it's monomorphic. There's a polymorphic site. There's a monomorphic site. So in this, I've got 2 segregating sites. So for example, this is a singleton, right? So in this site, I've got a single copy. The other question is, which is the ancestral copy, which is the derived copy? This is the idea about polarization. Some tests require it, some tests don't. How do you polarize the data? You look at an outgroup. Look at, for example, this is, so if you look at this sequence here, you might imagine, well, A's got to be the new mutation, but suppose that's in humans. Let's go back and look at orangutan. Orangutan is actually fixed for A. That tells you that C is the new site. So in site frequency data, you often distinguish between an ancestral allele, that is the allele present before the mutation, and the derived allele, that is the new one. So here, whether A or G is ancestral derived, whether C or A is ancestral derived, you can't tell just from the sequence. You need to polarize that data by looking at an outgroup. There are a lot of tests that don't worry about that, but there's some tests that actually use that. So for example, under selection, you should see an excess of rare derived alleles. So you can analyze this data as a series of haplotypes. We call those alleles. Or you can analyze this data as a series of sites. Infinite alleles, infinite sites. The allele frequency spectrum, I've got 5 alleles, 5 sequences here. I've got 1, 2, 3 alleles. So in the allele frequency spectrum, N1 equals 1. I've got 1 sequence as 1 copies. N2 equals 2, because I've got 2 sequences. In a site frequency category, I'm not going to polarize it. So 1, 2, 3, 4, I've got 4 copies which are monomorphic. So if I basically call that 5, so N5 equals 4, then I've got 1 copy where I've got 2 of one and 3 of the other, and then I've got 1 copy where I've got a singleton. So I can have allele frequency spectrum, and I can have site frequency spectrum. And again, we'll get back and talk about all this, but this is to kind of set the framework. So people see you can take DNA sequence and analyze it basically 2 different frameworks. Yeah?
Can you explain what's the phase data?
Yeah, good question. So basically phase data is, suppose and again, thanks for asking questions like that. Suppose your data is this. I tell you that you've got A, C, G. And the underline, those are your pairs. So basically, at one locus you've got that, at the second locus you've got that, at the third locus you've got that. So from a site standpoint, it's not a problem. From a phase standpoint, is this AAC? Is this ACG, right? phasing basically is if I just simply tell you what the heterozygote state of the data is, that doesn't tell you what the haplotypes are. So another way to think about it is, suppose I tell you you've got a big A, big B, little b. Well, that's either this phase or this phase. Those are different things. I can analyze this with site frequency spectrum, but here, allele frequency spectrum, I have to know, is it this configuration or is it that configuration? So allele frequency spectrum requires you to phase the data, which is sometimes a limitation. There are programs that phase the data for you statistically. I think those are a concern when you're doing statistical tests for selection, because they basically assume Hardy-Weinberg. So if you're going to phase data, you actually have to do physical sequencing to phase it. Right? So site frequency spectrum requires potentially polarization. Is that a new mutation or is C the new mutation? You can do it with an outgroup. Allele frequency data requires you to phase the data. And here are the data that they are phased. So if you have allele data, one of the early classic results was a result by Warren Ewens. There's Warren, Australian, University of Pennsylvania. And in the 70s, Warren came up with something which other people called Ewan sampling formula. It's kind of crass to come up with something and call it like Bruce's sampling formula. You throw it there and let people name it after you. And the cool thing is what Warren showed was you can describe the entire features of a sample from neutrality with one parameter, theta for any mu. So what this basically says is, what's the probability if I take a sample of size n that I have k distinct alleles? That's very powerful because you can then ask, if you know the value of theta, do you have too many or too few alleles in your sample? This was the first formal test using marker data for selection. And what you can basically show is you get this expression here. You basically get, don't worry about the L, that just means you're accounting it for the length you're looking at. Theta to the k, and these things here are called Stirling numbers. And those Stirling numbers are defined by basically either this polynomial or coefficients in that polynomial. The bottom line is that Warren came up with a formula that says if you give me the number of sequences you sampled and an estimate of theta, I can tell you the expected number of alleles. And you can ask, is that number of alleles too many or too few? The number of alleles is the crudest measure of the frequency spectrum. A more exact measure, so for example, you can ask, what's the probability you're monomorphic? That is, you have one allele. There's the expression. You can ask about the mean and the variance of that. So the nice thing about Ewan's sampling formula was that if you know theta, right away you can take a DNA sequence and ask, do I have an excess number of alleles? Do I have a deficiency of number of alleles? And again, you can ask if that's statistically significant. The problem, of course, is this doesn't just require neutrality, it requires mutation, drift, equilibrium. And you could have neutral alleles, but not mutation, drift, equilibrium, and observe way too many or way too few alleles based upon this sampling formula. Yeah?
What did you use as formula? It was no sequencing data in the 70s.
Smart guy.
Okay.
He anticipated it. So what they used wasn't sequencing data, they used highly polymorphic allozyme data, so protein variation. But remember, so when did Watson Crick come up? 50s, right? 53, I think. 64 is the infinite alleles model of Crow and Kimura that basically said, take a DNA sequence, each new mutation is unique. That was way before DNA sequencing, right? So one of the really interesting things is population genetics has kind of anticipated where the technology is going to go. A result I'll talk about a bit later on, something called the Hill-Robertson effect. When we look at genomes now, Hill-Robertson is how we use to basically analyze genomes. Hill-Robertson basically says, if I have selection at a linked site, it's going to make selection at a nearby site much less efficient. That is, it will make it more neutral. Hill and Robertson were animal breeders. They came up with this notion in the 60s. long before anyone thought you could estimate localized levels of D, and yet it's now the thing we use to analyze genomes. There's a lot of really interesting things in population genetics out there, which fortuitously kind of saw where the technology is going to go. I don't think they had great insight, I just think they had generalized results, and as technology caught up to the results, they could see. So anyhow, people did apply this because, remember, let's frame this. So in the 60s, you had the experiments by Lewontin and Hubby, and you had this great error called find 'em and grind 'em. And what people would do, when Lewontin and Hubby figured out you could separate protein variation on starch gel electrophoresis, people thought, let's take Drosophila, grind them up, and we'll run them out, take that grind extract, run them out on a gel, and stain with things that make esterases stain. And they saw esterase protein and thought, wow, let's try another thing. And then works with Drosophila, I'm hungry, let's do lobster, right? So there's this huge explosion of papers where people went out, sampled their favorite organism, ground them up, and ran them on a gel and stained it. There's a massive explosion where people saw too much variation. And the point about Lewontin and Hubby was the model going in was people thought there wasn't going to be that much variation because people thought it was all mutation drift. People thought it was all balancing selection or removal of deleterious alleles. They expected small numbers of variation. They found far too much variation than they expected, and it put the field in a bit of a frenzy for a while. The neutral theory eventually came out of that frenzy. And so there was a lot of actually allozyme data at the time that people were concerned with. Are there too many alleles in this sample given what we know? So even though we now apply it to DNA sequence data, originally it was applied to allozyme data. Yeah?
Question about data. So is that how long will it take for you to get to this one copy given the mutations? That's sufficient.
So let me see if I understand your question. I think you're asking about singletons. You said get to one copy?
No, I mean the parameter that is important here is theta, right?
Sure.
What situation is that?
So you're asking a different question. How can I estimate theta, right? So there's a bunch of things we can use to estimate theta based upon other observations that we make. So remember, when I take a sample, in that sample I can estimate, for example, levels of polymorphism. I can estimate nucleotide diversity. I can estimate number of singletons. All those give me independent estimates of theta. So we'll talk in a second about how we get theta. For now, UN sampling formula says if I had some way of estimating theta, I could then get these results here. The second question is, how do we estimate theta? Let me get that question in just a second. It's a good question. So let me just flesh out a couple more things. We'll get to estimating theta. You can likewise, Warren actually worked out the full allele frequency spectrum. So given an estimate of theta, given an estimate of number of sequences, not only how many alleles do you have, but how common are they? How many are present in one copy, two copies, etc. This is the full allele frequency spectrum, and it basically says, given an estimate of theta and your sample size, I can actually write down an expression that will tell me not only how many alleles will be there, what their expected frequencies should be. So how do I estimate theta? Well, let's go to infinite sites. We're going to talk about a nucleotide diversity, number of segregating sites. So nucleotide diversity is basically this. I take two sequences. I ask, how do they differ, right? I count those up, and you can basically get an estimate of theta by basically taking, this is called the Tajima Estimator. You can basically look at the average number of pairwise differences among two sequences, standardize it, and get an estimate of theta. So here's one estimate of theta you can plug back into here. It turns out there are other ways to estimate theta. I can look at the number of segregating sites. So I look at my sample, not segregating, not segregating, segregating. So in my sample I've got 5 segregating sites. I can then estimate what theta is based upon the number of segregating sites, and this correction factor is determined by the sample size. We'll see where all these come from a little bit later on today. So if I have the nucleotide diversity, I can use that to estimate theta. If I have number of segregating sites, I can use that to estimate theta. And the key about that is those are two different estimates of theta. They should estimate the same theta. Tajima's D-test asks, are those estimates consistent? So a lot of the molecular tests are based upon different ways to estimate theta, which we'll talk about in a lot of detail. So the key feature here is the importance of Warren's paper is it was the first paper that basically said, can I give you a description about what that sample should look like if I know the sample size and I know theta? And Warren said, yes, if you know theta and the sample size, here's the expected probability for the number of alleles. And furthermore, here's not only the number of alleles, but here's the frequencies I expect those alleles to be. And so if you have an estimate of theta, you can basically ask, is that consistent with it or not? And furthermore, if you condition on k, you notice what doesn't appear in here? What's not in this expression? So this means if I tell you what n is and k, what's missing from this expression? What Greek letter don't you see? There's no theta. When I condition on number of alleles, I don't need to know theta. So Ewan's formula says, if I condition on the number of alleles, under neutrality, theta goes away, because k is a sufficient statistic for theta, and then I can actually tell you what these are. So this version of Ewan's sampling formula says I don't even need to know theta. This is entirely based upon the sequence. If I condition on the number of alleles I see, this gives me the expected frequencies I should see those alleles at. That gives you a clean test, independent of having to know theta. You can also ask if the number of alleles is excessive by having some other estimate of theta. Again, these are things we're all going to talk about in detail. The bottom line is, with either allele, the infinite alleles model or the infinite sites model, if you have the value of theta, you can describe a number of features about that sample. You can then turn that on its head. You can use a number of features about that sample to independently estimate theta and ask, are those independent estimates consistent with each other? You expect them to be consistent under drift. You don't expect them to be consistent under selection. And again, we'll talk in detail about that. So what's the important points so far? I'm setting up the background for the kind of molecular bookkeeping we're going to have to do. So one critical thing is this notion of the standard equilibrium model. If I've got neutral alleles, I can predict what the pattern is going to be of variation when I'm at equilibrium. That is, the rate at which new mutations are introduced exactly is balanced by the rate at which they're removed. So the standard neutral model, also called the neutral equilibrium model, assumes neutral alleles at drift mutation equilibrium. That's a big assumption, usually violated. Under that model, a large number of features of the sample, how many alleles, what their frequencies are, how many appear as one copy, what the average number of differences, a number of features are entirely defined by theta, 4ne mu, and the sample size. We can look at that data in two different perspectives. Number one, I can look at those as a series of alleles. That's a whole DNA sequence. Number two, I can look on a site-by-site basis. One is called infinite alleles framework. One is called infinite sites framework. So after our break, we'll come back and talk a bit more about that. Then we'll talk about what happens when you have selective sweeps. Again, what I want to stress is there's a lot of details here. I'm just trying to show you where it comes from. Don't get bogged down in the details, because we're going to come back and unpack all these at length. Remember, this is the first introductory lecture of stuff we're going to cover for the next 2 days. So this is kind of setting the basic ideas: infinite alleles, infinite sites, neutral equilibrium model, singletons, segregating sites. We're going to come back and flush all those out over the next couple of days. Okay? So we've moved from quantitative genetics, where we care about effects of alleles and phenotypes, into entirely population genetics. We're simply looking at sequence variations and looking at frequencies of those sequence variations. Okay? So let me go back and remind you of a couple of key points. Then we can, whoops. I hate this silly ear thing, but they say it's the most efficient. So when you've got, what we're looking at basically is if I take a sample of sequences, what can I say about its features, number of alleles, polymorphisms, etc., under the standard equilibrium neutral model. So I have neutral alleles, and the population size has been constant long enough, so I'm at an equilibrium between drift removing alleles and mutation pumping in new alleles. And key point number one is that most of the features can be described by this parameter theta, which is 4 times the population size times the mutation rate. If I'm looking at a haplotype, the mutation rate is for this entire sequence. If I'm looking for a site, the mutation rate is for each individual site. Then if I have data, I can treat that data in 2 different frameworks. Framework number 1, the so-called infinite alleles or infinitely many alleles, is I can consider basically each haplotype as an allele and ask how many different haplotypes there are. There's 2 assumptions that go into that. Assumption number 1, we don't have any recombination in our sample, so new variation is generated not by recombination, but simply by mutation. So if my DNA sequence is small enough in your sample, it's not a big deal. Number 2, the infinite alleles assumption is each mutation generates a new allele. Even in a sequence this small, the odds of a single mutation back mutating, so if any of these can mutate, the odds of back mutating and get this allele are extremely small because it means that none of these mutate, and this only has a 1 in 3 chance of mutating back to here. So even in a small sequence, the infinite alleles assumption model is quite reasonable. Infinite sites say I can, instead of having the sequence, I can then look at this that is a series of sites. That's a monomorphic site, that's a monomorphic site, monomorphic site, segregating site, monomorphic site, segregating site. So in this analysis, if I treat this as infinite alleles, I've got 1, 2, 3 alleles. 1 allele is present as 1 copy, and 2 alleles are each present as 2 copies. If I look at it from an infinite site standpoint, I've got 1, 2 segregating sites, and one of those sites is a singleton. Now, the minor caveat is when I go to the infinite sites model, sometimes I care about which allele is ancestral and which allele is derived. Derived simply means I have a new mutation. So for example, here you might think, well, A is the derived allele, but it could be that the mutation is actually C. So in order to polarize or determine which allele is the derived allele, I basically need to look at an outgroup. So for example, if this is Drosophila, I then look at a sister Drosophila, I see that in that sister Drosophila, everybody is fixed for A. Well, that tells me that C is a mutation. Or if everyone's fixed for C, it tells me A is a mutation. So if I've got site frequency data, sometimes I polarize it, or I could not polarize it. If you don't polarize it, it's simply the minor allele frequency. So here, the minor allele frequency is 2/5, here it's 1/5. And you see, as we'll talk about those both ways. One's called the folded frequency spectrum, where I focus on the minor allele frequency. The other is called the unfolded. We'll come back and talk all about that. Let me see a couple more things, review, and then we'll see if there are questions. So if I have the infinite alleles framework, the summary statistics that make the most sense initially are, number 1, how many alleles do I have? And number 2, what are the frequencies of those various classes? So for example, if I've got 5 alleles, are they all present as 1 copy? Are they all present equally? And the key is that Warren Ewens came up with something called the Ewens sampling formula. Of course, he didn't call it that. He called it the sampling formula. And what that shows, if you know theta L, theta L simply is the mutation rate for this entire sequence here. So simply sum up all the mutation rates. If you know that, then if I give you the value of that and give you the sample size, you can compute the probability of seeing 1 allele, 2 allele, 4 alleles. That right away allows you to ask, is the number of alleles too many or too few? But Warren went a step further and noted that if you condition on, so this is the number of alleles present as 1 copy, number present as 2 copies, number present as n copies. If you condition on the number of DNA sequences and the number of alleles you see, theta goes away. So K is what's called a sufficient statistic. K contains all the information on theta. So this expression here basically allows you, given your sample size and the number of alleles, to ask if this configuration of the frequency of those alleles, given the number of alleles in the sample size, if this configuration departs significantly from neutrality. So in an infinite alleles framework, we mainly ask questions about how many alleles are there and what are the frequencies of those alleles. If we then move to an infinite sites framework, there's several other statistics we have, and each of those statistics, its expected value is completely determined by theta, 4 times the effective population size times the mutation rate for that site. And since its expectation is determined by theta, we can flip that around and use these different estimates to actually estimate theta. And a mini-test asks, are these estimates consistent? So for example, Tajima's D-test, which a lot of people probably heard about, asks if the estimates of theta based upon nucleotide diversity is consistent with the estimated theta based upon number of segregating sites. So let me just say a couple more things and open it up for questions. So nucleotide diversity is quite simple. I take 2 of my sequences, I simply count up all the number of pairwise differences between them, and I compute the average of that over all combinations of sequences. This is essentially a heterozygosity measure. And the nice thing about that is this is our estimate here for nucleotide diversity. Nucleotide diversity exactly equals my estimate. The expected value of nucleotide diversity is theta. So one estimate of theta in infinite sites is the nucleotide diversity. That's often called the Tajima Estimator, because Tajima used it in his test. Another estimate is the number of segregating sites. So I count the number of segregating sites, and I have the L over here for the length. This gives me the per site mutation rate. Number of segregating sites, and then basically adjust it for this thing here, which is entirely a function of the sample size. So if I look at nucleotide diversity, under the neutral equilibrium model, I can use that. The expected value of nucleotide diversity is theta. I can look at number of segregating sites, make this adjustment for the length and the sample size. That then gives me another estimate for theta. Tajima's D-test asked, are those 2 estimates consistent? And then I can also look at the full distribution of sites. How many sites are monomorphic? How many are present as 1 copy? How many have 2 copies, 3 copies, 4 copies? I'll get that in a second. That's the site frequency spectrum. So over here, the allele frequency spectrum says, how many alleles do I have, and how many alleles are present as 1 copy, 2 copies, 3 copies, 4 copies? The site frequency spectrum says, I go down each site and ask what fraction of sites are monomorphic. What fraction of sites contain 1 derived and the rest ancestral? What fraction of sites contain 2 derived and the rest ancestral? 3 derived, 4 derived, etc. So the same data I can look at in terms of frequencies of alleles or frequency of different types of sites. So with that background, one of the comments was, what about sequencing errors? And sequencing errors are an issue. What would be the consequence of sequencing errors? You'd see more singletons and you'd see rarer alleles. So there are methods we'll talk about which basically remove singletons and ask for that. The other issue is that usually people use sufficient coverage so that sequencing errors are not thought to be much of a problem, or if they're concerned about it, they go back and they resequence an area and get higher confidence in there. That kind of takes you back to where we are. Again, the big picture for all of this is I'm giving you some of the basic terminology and jargon and ideas we're going to be using over the next 2 days for formal tests of departures of the neutral equilibrium model. The neutral equilibrium model can show a departure because you don't have neutral alleles. However, it can also show a departure because you have neutral alleles, but they're not at equilibrium. So a big concern is, how do we adjust for the fact that we can get false positives when we're not at equilibrium. And one key idea is that if you have demographic forces acting, they should act roughly equally on all the sequences in the genome. So there are a lot of ways, many simply ad hoc or heuristic, that people use to try to account for that. For example, compute a whole bunch of FSTs and look at the outliers. Those issues, those methods have problems, we'll talk about those. But the basic idea is we're introducing some of the infinite alleles, which basically allows you to look at disequilibrium, but requires phase data, and infinite sites, which simply require sequencing data, not phase. Those are kind of the basic raw information we have then to plug into this wide variety of tests we'll talk about. Okay. That kind of takes us to where we were. I realize it was a bit hard slogging this morning, but I want to let you know that we're going to come back and introduce these concepts over and over again. I wanted to kind of introduce them once to kind of set it in your mind a little bit. Yeah, sure.
For all those tests that you're seeing now, is Hardy-Weinberg big assumption?
That's a great question. You don't really, for most tests, you don't need Hardy-Weinberg assumption, because basically they're not looking at assumptions about, so Hardy-Weinberg basically comes in two flavors. Flavor number one is the genotypes can be expressed as allele frequencies. Deeper flavor number two, allele frequencies don't change over time. We're not worried about flavor number two. We expect that's going to fail under drift-in selection. Flavor number one is simply that the genotype frequencies are in the values we expect from allele frequencies. Well, almost all the tests look at alleles. They don't look at genotypes. So Hardy-Weinberg goes out the door there. We don't really use it. Yeah?
Does flavor number one correspond to essentially random mating within that generation?
So yeah, basically means random unit of gametes in the previous generation, and we were sampling them before selection has perturbed that. So don't get, so do people know about Hardy and Weinberg? No, about Hardy and about Weinberg. Weinberg was a German physician, and he was one of the first people to actually get the concept of ascertainment correction bias. So he was way ahead of his time. But he's not the most interesting person. Hardy is the most interesting person. Who was it was keeping track of their hours in academia? Someone was? There you go. You need to read Hardy's book called A Mathematician's Apology. It's basically a mathematician says in your face. And Hardy was a very famous number theorist. A lot of number theory traces back to Hardy. And Hardy's typical day was he would get up. Of course, he'd sleep in a little bit, maybe get up at 8 o'clock, 9 o'clock, have a nice breakfast, take a nice walk, go into his office, work for about an hour, have lunch, play squash, go back to his office about an hour, go home, and repeat. And Hardy was incredible. This is what you can hope for if you're a tenured professor, right? Hardy was extremely pleased about the fact that nothing he did had any practical relevance. He worked on number theory. So when a colleague of his, Yule, came up and posed a problem, and the problem was when Mendel was rediscovered in the 1900s, the concern was, all this dominance. By the way, just a quick aside, for those of you that are going to be teaching genetics, let me tell you a big assumption you give your students without realizing it. When you introduce Mendel, all 7 of Mendel's genes are what? Fully dominant. So students assume that genes are always dominant or recessive because they see 7 examples. So when you introduce Mendel, you need to stress that Mendel's were outliers. Most genes don't show complete dominance. Mendel's were exceptions. He could score easy. But Yule's concern was if genes have dominance, why wouldn't the dominant gene take over the population? When it's present as the heterozygote, you only see the dominant phenotype. So he went to his mathematician friend who realized that no, it's just the allele frequencies don't change. And Yule convinced him to publish this. But if you're a famous mathematician who's extremely proud of the fact that nothing you did had any practical application, where would you publish such an article? In this backwater American journal called Science that no one would ever read. Now, the real irony is, what's the most heavily funded branch of mathematics by the National Security Agency and by big companies? number theory, because that's encryption. So Hardy was so proud of his stuff having no application whatsoever. It's probably the most applied branch of mathematics around in terms of, big implications for security and finance and stuff. So that's Hardy-Weinberg. I think it's always fun to put names with people. Science is very much a public enterprise. Science is heavily influenced by politics, not necessarily one party or the other, but strong personalities can often corrupt a field. And a benevolent personality can often make a field wonderful. So we often have this view of science as kind of being iconic and covered with golden light and, where human frailties don't come in. That's complete Bravo Sierra. Science is very much human enterprise. It has self-correcting mechanisms to eventually get its act together, but it'll often take a while to do so. And finding out about some of the personalities, I think, makes science a bit more interesting and gives a bit more flavor and color for it. So you'll see a lot of pictures as I go through and talk about various people. Other questions or comments or clarifications? Again, I want to compliment you on asking questions. I especially appreciate, like when I said phase, I especially appreciated the question to ask to clarify that. So, any jargon comes up or you're not quite sure what I mean by something, do not hesitate to ask. You've been really good, but I want to encourage you to continue to do that. Yes? Sure. So how nucleotide diversity is linked to heterozygosity. And the easiest one to think about is imagine I'm simply comparing one site. So if I'm a heterozygote, I score a difference. If I'm a homozygote, I don't score a difference. That's kind of the quick way to think about that, right? So if I'm looking at a single site, nucleotide diversity is heterozygosity. If I'm averaging over a bunch of sites, it's essentially the average heterozygosity over those sites. Is one way to think about that. But again, the key feature here is I've got this measure I can easily compute from sequence data, and the expected value of that measure equals this parameter called theta. So if you ask, what's theta? If you assume the neutral equilibrium model holds, you can take your DNA sequence data, compute the nucleotide diversity, and the expected value of that is theta, so your estimate of theta is the nucleotide diversity. So for example, you could have a scanning window of nucleotide diversity as you go across the genome. And you can see how it changes. Part of that change will be because local mutation rates vary over genomes, right? Some sequences are hypermutable, some sequences are really cold when it comes to mutation. The other thing we'll see about is selective sweeps strongly influence that. So all these basically here are assuming that you have neutrality and it's only influenced by neutrality and nothing else. Other? Yes, please. So polymorphism come in a couple of ways. Probably the simple way is number of segregating sites. If I look at sites, what fraction of those sites are polymorphic? If I look at L sites, S over L is the fraction of sites in my sample which are polymorphic. It doesn't say how polymorphic they are. Nucleotide diversity tells me that. So for example, it could be that each of you is a site. So you're a site, you've got this big stack of 50 alleles in you. It could be that every site is polymorphic, right? But it could be that every site is polymorphic because every site has a singleton. In that case, nucleotide diversity is rather low. So these are different measures. And the nice thing is, under the neutral equilibrium model, they're all connected by theta. So they all have interconnected expressions if you have the neutral equilibrium model. If you don't have that, then the values are expected to differ. So number of singletons, number of segregating sites, nucleotide diversity, those values may be at odds with each other. That is, they give you quite different estimates of theta if the data are perturbed by selection. And so a lot of what's called site frequency tests, Tajima's D, Fuin Yu's Wei, and a whole bunch of other things, those are all basically looking at contrasting different estimates of theta using different parts of the data, that is, different parts of the so-called site frequency spectrum. We'll have a whole lecture, an exciting whole lecture just on that. But for now, just trying to get infinite sites, infinite alleles. the notion of diversity, notion of segregating sites, the notion of a frequency spectrum, a little bit odd, but it's simply a way to categorize the frequencies of alleles or the frequency if I look over sites. And the key is all those can be related to the sample size and theta under the neutral equilibrium model. So tests of selection look for departures from that pattern. Tests of selection are basically based upon usually contrasting two things. One observation gives me this estimate, a second observation gives me that. Are those two observations compatible? If they're not compatible, you reject the neutral equilibrium model. People get excited, thinks you're rejecting neutrality. Usually you're simply rejecting equilibrium. And we'll talk at length about how you distinguish between those two. So this is the big sticky world that is tests of selection using molecular data. There's a gazillion tests. Everyone has their own test. And all these tests have problems the creators don't want to really tell you about. My job is to be a speaker of truth and to let you know why they work and don't work. So when you apply them, you can apply them with some open-eyedness, and so you won't get comments back from the viewer you don't really expect. So I got to think about 3 more slides on this, then we'll get into what happens with selective sweeps. So the site frequency spectrum. So let's go back and ask what that site frequency spectrum is. The allele frequency spectrum simply says, here's my sample. I don't really care about, the alleles have no special feature one versus the other. So in the allele frequency spectrum, I simply ask how many alleles are present as one copy, a singleton, how many present as 2 copies, 3 copies, etc. That's the frequency spectrum, the allele frequency spectrum. The site frequency spectrum says, okay, now my sample is, let's look at each site. So how many sites are present such that all the copies are the ancestral copy? Again, you've got to polarize it. So 1 site, 2 site, 3 site, 4 sites. So 4 sites all have the ancestral copy present. I've got 1 site, let's say A is ancestral, that has 2 copies of the ancestral. And I've got 1 site, let's say that's ancestral, that has 1 copy. So you go by site and simply look at how many sites have 1 derived, 2 derived, 3 derived, 4 derived, 5 derived. That's the site frequency spectrum. And that site frequency spectrum can be folded or unfolded. Folded, well, let's go unfolded first. Unfolded, it ranges, if my sample size is, let's say, 100, then it ranges from 0 to 100. And it says, at a site, how many copies do I have of the derived allele? That is the allele that is mutated. How do I know it's the derived allele? I need an outgroup. Not a big deal now, but it was a big deal in the early days. That's the unfolded frequency spectrum. The folded frequency spectrum is what? You fold it about a half. And you simply ask, how many situations is the minor allele present as 1 copy, 2 copies, 3 copies? Since it's the minor allele, it can never be more frequent than half the population. So in the folded frequency spectrum, I take that frequency spectrum and I fold the value from a half to 1 back over on itself and double count. But that doesn't require any polarized data, right? I simply look at the minor allele frequency and ask that. The folded frequency spectrum is used in a lot of tests, but the unfolded frequency spectrum actually has a bit more power. So folded frequency spectrum, site frequency spectrum, simply uses the minor alleles. And we can look at the site frequency spectrum in 2 different ways. Number 1, as a feature of the entire population. Number 2, as a feature of a sample. So I could ask, in the entire population, what fraction of sites, X, have a particular allele frequency? So for example, in that entire population, what fraction of the sites have 40% derived allele frequency, or in an unfolded frequency spectrum, or 40% minor allele frequencies? So I get a distribution that ranges, X ranges from 0 to a half in a folded, 0 to 1 in an unfolded. That's for the population. Then I can ask for a sample. What's the number in a sample that have K derived alleles or K minor alleles? depending upon folded or unfolded. And the answer to both of those is something called the Watterson Distribution. Jeff Watterson, he worked with Warren Ewens. I actually couldn't find a picture of Jeff. I thought I had one. So in the Watterson Distribution, the unfolded Watterson Distribution says the frequency of sites where the derived allele is X is given by theta over X. So what that looks like here, if that's 0, that's 1, it falls off like that. And what it says is most sites have tiny derived allele frequencies. And it falls here because this says 1 copy is present. This says all copies but 1 are that. That's for the population. There are a number of tests that basically ask how that number is perturbed when you have a sweep. Again, this, notice it depends upon theta. This result here, again, is an equilibrium neutral assumption. The folded frequency spectrum basically gives you this, because that's the value from 0 to 1/2, that's the value from 1 to 1/2. So the frequency spectrum, if I now base it not on the derived allele frequency, but the minor allele frequency, gives you this expression here. So unfolded and folded, this says in the population, what fraction of sites do I expect to have a frequency of 20% or 5% or 4%? That's a feature of the entire population. If we ask for the feature of a sample, the Watterson distribution has a very similar form, except now it has i's. This says what fraction of your sample has I copies. That is, has I derived alleles, this is the unfolded, or has I minor allele frequency alleles. And we're going to come back and use these expressions when we develop tests for selection. So let's look at the big picture and not the details. The big picture is if I have a sample, and in that sample I count up how many sites contain 1 derived allele, or 2 derived alleles, or 3 derived alleles, or 1 copy of the minor allele, 2 copies of the minor allele, 3 copies, so unfolded and folded. I have a pattern what that should look like based upon the equilibrium neutral model. I can then use that and ask, is the actual observations I see, do they depart from that pattern I expect under equilibrium neutrality? All these tests using marker data, I first of all have to have some expectation of what neutral data looks like, and then contrast that to my actual data. That neutral data depends upon if I'm looking at infinite alleles or infinite sites framework, but in both cases, the key parameter is this parameter theta, and so if I know theta, all these different measurements I can describe. Number of alleles, number of segregating sites, number of singletons, nucleotide diversity, are all connected through this parameter theta, so those should all behave in a specific coordinated fashion. Tests for selection ask, do some of those estimates behave as not expected? So if one is big, the other should be big, we see it small. So a lot of tests basically are based upon departures from the allele frequency spectrum, the site frequency spectrum, or summary statistics. The allele frequency spectrum and the site frequency spectrum are the full data. The summary statistics like nucleotide diversity or segregating sites are summaries. So if I give you, I could have several different allele frequency spectrum that give you the same number of segregating sites, the same nucleotide diversity. So the full data is given by the frequency spectrums. Things like number of segregating sites, number of singletons, nucleotide diversity are summary statistics of that much broader site frequency or allele frequency spectrum. And that kind of lays the foundation. We're now going to come back after we talk about sweeps and start using that. Yeah, thank you. Appreciate the heads up. Right, so for example, the site frequency spectrum, the unfolded, looks at the frequency of derived alleles. A lot of times you don't have that information. Let's say you're working on a non-model organism. You don't have an obvious close outgroup, or you don't have, you know an outgroup, you don't have funds to sequence it. You can use the folded, which then simply asks about the minor allele frequency. It gives you information. There's slightly more power in using the unfolded, but for example, Tajima's D test uses the folded frequency spectrum. So there are a lot of tests that don't require the unfolded frequency spectrum. Yeah, and by the way, if you're on my peripheral vision, I don't see you, and you had your hand up, it's okay to say question. I appreciate the heads up pointing that out, but if you're dressed in black so you kind of blend in there. And I also, when I teach big class, I have students that do this. In a class of 500, it's like, you know, and like they're like 3/4 of the way back in the class. Like, that's not really helpful. You know, are you doing this or are you asking a question? So if you had your hand up and I don't see you, just say question, because what I'll often do is I'll acknowledge that, finish a point, and then come back to you. It's, yes, it's normalized to get a probability distribution. Yeah. And there are a variety. So if you've heard, for example, the program Sweep Finder or composite likelihood ratio test, those are basically take the Watterson distribution and look and see how it's perturbed by a sweep and ask if the pattern you see is consistent with that. So the Watterson distribution actually underlies a lot of these tests, but it's critical because it assumes equilibrium. And the equilibrium assumption is probably validated far more often than the neutrality assumption. So a lot of things we'll talk about is how do we get around the fact that a lot of these early tests were based upon the assumption not just of neutrality, but of equilibrium. The population size has been constant long enough so the removal of variation by drift is exactly balanced by the input of variation by mutation, the equilibrium assumption. Other questions before we move on to sweep theory?